Okay, well, it really is the greatest possible pleasure to introduce our next speaker. He holds the chair of James Ault Professor of Psychology at the University of Michigan, named after James Ault, the man of the electrical stimulation of the brain, who was an academic at the University of Michigan. Kent has recently come out as one of the most influential living psychologists, so it's a particular honour to And I guess it was 84 at Sussex University. We were both at a conference, and then I met up with Kent again in 91. He'd just done the work with dopamine, with Elliot Fellenstein on dopamine, and he stayed over in my flat in Milton Keynes. We went to the pub, and he was telling me all this. My attitude was more or less, go away and sharpen up your pencil and come back. We all know dopamine is the hedonistic molecule. And then he said, well, actually, you know, we're basing our next review paper on your model, so, you know, being flattered, I became quite sympathetic. The idea of dopamine being this. Kent was a student at University of Pennsylvania, at Dalhousie in Nova Scotia, and University of Sussex. For some years now, he's been in the beautiful town of Ann Arbor, quite near to Detroit, Michigan. Absolutely delightful place. Now he's in the equally delightful place of Lewis in Sussex, while visiting professorship to Sussex University. I think I don't need to say any more because I've said so much about his work so far, but I'll just pass you over to Kent.
Thank you, Fred. It's a real pleasure to be here at an Open University event, and especially a pleasure to do this with Fred. Our careers have intersected going back all these years, and his theory really has turned out to be a beacon that's guided my career now for decades. And I'll say a little bit more about that as we go along. There's three things I'd like to do today. First, I'd like to recap some of the principles from the marvelous incentive theory that Fred described so well in detail before, but there's a few principles to distill, and I'd like to do that. So a brief recap of liking versus wanting, and just distill the principles of the motivation theory that we're going to use to understand some of the new phenomena. Then the second thing is to look at a couple of the new phenomena, some of the new experiments that we're seeing where we can really pull liking away from wanting more powerfully than we've ever done before, and even create wants for what hurts you. Sort of wants that are relevant to addiction, but wants that really give, I think, a key insight into the nature of the wanting process, the incentive motivation process that can be pulled apart from pleasure. And then the third and last thing is to look a little bit more at some of the clinical applications. Fred described a number of clinical applications in depression as well as addiction, but we'll look at a couple of specifically the wanting versus liking detachments that other people are applying and using today in clinical human situations. Maybe the first thing to say is that in my career, it's been a story of surprises and often disappointments. I think the universe is willing to whisper new truths to us in experimental evidence, but often, at least in my experience, the truths it whispers are not the truths we were looking for at the time. And sometimes they're actually truths we'd rather not be true at all. But if we listen to the whispers, sometimes we can learn new truths. So even 20 years ago, if you had asked me about the TOTS incentive theory and dopamine wanting versus liking, I would say it's very important for drug addiction and drug motivations, for foods, for sex, for basic biological, all of the first tier from Maslow and a few of the second tier rewards that we're motivated to pursue in life. But probably not so important for the top tiers of Maslow, for social love, for creativity, for cultural and creative aspirations. I would have been wrong, I think, now I think I would have been wrong 20 years ago to say that dopamine was involved only in the bottom tiers and not in the top tiers. because the last 20 years of neuroimaging studies have really shown that not only is food and drugs and sex activating the mesolimbic dopamine-related brain systems that are important for the incentive salience kinds of wanting and basic pleasures, not only drugs and sex, but also things like favorite music of music students at McGill University who are dedicated to music. Their lives will be in music. Listening to the music that gives them their favorite chills, the most pleasant music, lights up the nucleus accumbens, the same brain dopamine systems. And even something as abstract, a general sense of well-being, as abstract as a general sense of well-being. This is from Richie Davidson at Wisconsin, showing that older adults, to the degree that they report feeling confident and positive about themselves, a general sense of hedonic well-being, is correlated with sustained activity in their nucleus accumbens, this subcortical structure that's the same structure involved in wanting and liking drugs and sex and food, the basic pleasure. I wouldn't have expected that 20 years ago, but what it's telling us is, just as Fred said, evolution, once it created this system for basic sensory pleasures, it's now in our human cultural evolution become co-opted even by the higher pleasures, even by looking at the face of the loved ones or listening to one's favorite music. So, two things to say about that. The first is, it suggests that what we can learn from studying the basic pleasures and rewards, which we can study in animals, often will apply to humans because it's the same brain system shared. With sort of a generalization, the hypothesis is that the conclusions will generalize to cognitive ones. But the second point, as I leave the slide, is that all of these are cues. All of these experiments are actually following the Totes-Bindra model of incentive motivation. What are they doing? They're playing the favorite music. They're showing images of sex. They're showing images of drug paraphernalia. They're showing images of the tasty foods. They're catching lives. They're asking people to reflect on their life at the moment and to visualize the state of their life. These self-generated imagery cues and physical visual cues that are presented in neuroimaging studies, these are the predictive cues that tap into the dopamine prediction error system. These are the cues that trigger wanting, and they're the way the system works. This is the Bindra-Totesian system. It jumps back to Dalbert Bindra, actually, whom I had never read until I read Fred's book. But Bindra, who died in 1978, had argued all along it was cues that were really guiding motivation, Pavlovian cues, CS pluses, that gained incentive value of whatever rewards they were paired with. And he argued that it was the cues themselves that triggered the motivation, that was sort of the focus of the motivation, that was so often the powerful motivating factor. He was arguing against drive, aversive drive theories, like compulsions that Fred described so well. And he said that when we perceive these cues, it's actually transforming, our brain transforms their representation. Those neuroimages lighting up the nucleus accumbens is actually transforming the perception of the music, transforming the perception of the sex or the drugs or the other reward cues. It transforms how we experience those cues so that they grab attention, they become attractive as motivational magnets, they trigger peaks of wanting to consume their associated reward. All of this was Bindra. Bindra, he published this several times in the 1970s. It culminated in a paper just before his death in Behavioral and Brain Sciences in 1978. Some of you know Behavioral and Brain Sciences is a journal that allows invited commentaries from peers in the field. And there were many, many invited commentaries on Bindra's article, and they tore his article apart. Virtually every one of the peers who commented criticized Bindra's theory. Almost all of the criticisms were, in my opinion, not very legitimate. But it sort of shows the sort of face of opposition that Bindra faced. I hadn't actually read that paper until Fred drew upon Bindra's work. And Fred did something new with Bindra's work that had never been done before, which actually made it work much better. It said here that it's triggering peaks of wanting to consume the reward. That's often true. food cues can make us want to eat, but not always true. If you've just recently eaten, food cues don't make you want to eat. That's one of the few valid critiques that peers were raising against Bindra's article in 1978. Bindra, I think, was just sort of overcome by the array of opposition to him, and he never properly addressed that one valid critique. But Fred did. Fred, in his 1986 book, took the Bindra notion, allowed all of the Bindra rules to operate, but he also allowed cues to be modulated in their power from moment to moment by relevant physiological states. Are you hungry? Then food cues will become very potent at triggering wanting. Are you thirsty? Then water cues. In other states, sex cues. In other states, drug cues. Take a drink, and drug cues might become more potent at triggering the wanting. Homeostatic signals, physiological signals, could modulate together incentive salience and control both wanting and liking together to control all of the Bindra-Totzian features of elicited incentive behavior. This was the book that drew this all together and also laid out this system in parallel with a cognitive desire system to acknowledge that we all have cognitive desires, but to say that they could operate by different rules, and the rules that control this mesolimbic wanting system will sometimes pull us in opposite directions from the rules that control our cognitive desires. So we can have temptations we'd rather not have. That's very important. Fred mentioned that we met in 1984, and I remember our meeting. It was at this conference at Sussex University in the biological building, Maynard Smith building today, just outside the amphitheater, the lecture theater of the Maynard Smith building. And after some talks, we had all lined up for tea, and I was in line waiting to buy a cup of tea, and someone tapped me on the shoulder. It was Fred Totes. And he said, didn't you do a taste reactivity study? He says, I'm writing a book on motivation. Didn't you do a taste reactivity study on liking and disliking simultaneously? Now, I was 1 year out of graduate school at that time, at this meeting, and some of my close friends in graduate school had read some of my papers, and I had sent papers to my parents, but I don't think they read them. Fred was the very first person I had ever met at a conference who said, I've read your papers. So naturally, I responded positively to that. We had tea, but then later on in the afternoon, we went over to the art center where they had the bar, and we, I think, had a pint, and we talked about his book and incentive motivation and various things. And 2 years later, this book came out. Now, by that time, I was at the University of Michigan as an assistant professor, just starting, teaching my own classes in biopsychology of motivation, and so I used Fred's book as a textbook for the course. And in the beginning, the first couple of years, this book was simply one way to look at motivation, sort of useful for the students. But as the years went on, sometimes the students in my course would suggest new experiments to test the ideas, the Bindra-Totzian ideas in this book. As years went on, this became more and more a prescient, accurate, truth-predicting theory, that just made many, many accurate predictions in advance of the experiments. They predicted what would happen if we did the experiments, how cues would become powerful at triggering the motivation, how we could modulate the power of cues in triggering the motivation, again and again. So I became gradually a fervent believer in the Bindra-Tots model of motivation. And when we split it into liking and wanting, took the Tots diagram and just split it this way to have wanting here, this came to me initially, it was both a surprise to me and a disappointment originally, this original split of dopamine as wanting. At the same time I was teaching Fred's book in those early years at Michigan, I was also teaching that dopamine was pleasure. We all knew then that dopamine was pleasure. There was beautiful evidence that dopamine was pleasure. I believed it, and I would teach it to my students at the same time I was teaching Fred's theory. And when my colleagues Terry Robinson and I, and Elliot Nelson and I started to do taste reactivity dopamine studies, we expected it to be pleasure-liking. And it was disappointing that in my hands it could never be pleasure-liking. Disappointment after disappointment. Originally, the split of wanting versus liking and assigning wanting to dopamine, originally that was just a post hoc possible way of making sense of the first experiments when dopamine abolished all apparent wanting but left the liking reactions intact. Gradually, as we went on, turning on the dopamine system, it became clearer that, well, there was something to this split, and that, yes, we could control wanting power by turning on and off dopamine. So gradually, I came to believe that dopamine was wanting versus opioids were liking, and that Pavlovian cues could trigger the wanting, and that various states, including drug states, could modulate these wanting. In the 90s, my colleague Terry Robinson, at the same time that we were splitting wanting and liking and finding dopamine was wanting, Terry Robinson in his lab was studying the effects of drugs of abuse on brain dopamine systems. Everybody knew, even then, that drugs of abuse would turn on dopamine when you take the drugs. Cocaine, amphetamine, heroin, morphine, alcohol, nicotine, virtually all drugs of abuse will turn on these dopamine systems when you take them. But what was only just becoming clear was that the same drugs could produce more long-lasting brain changes, not in everybody who took them, but in some individuals. Some individuals were vulnerable to this long-lasting change, and the change was neural sensitization. Neural sensitization of the dopamine systems meant that they became hyper-reactive to the drugs. Hyperreactive to the drugs. They weren't always hyperactive, the dopamine system. They were not hyperactive. If you looked at the dopamine system in a sensitized individual most of the time, it looked perfectly normal. But if you gave the drug again, it gave a much bigger dopamine response than the drug would have done the first time it was taken. It gave a much bigger dopamine response than the drug would elicit in other individuals who were not sensitized. This was drug-induced sensitization. And the most important thing about drug-induced sensitization was that if you stopped taking the drug, drug-induced sensitization did not go away when you stopped taking the drug. Other things, like drug tolerance, that goes away if you stop taking the drug. Drug withdrawal, that goes away within days or weeks if you stop taking the drug. But drug-induced sensitization didn't go away. If anything, it strengthened during a month or so of drug abstinence, and could last half a lifetime in a rat. And there's evidence it could last a year in a rat, which is half a lifetime for a rat. There's evidence that it lasts at least a year in human experiments, and addicts suggest that it last half a lifetime. It doesn't happen in everyone. There's enormous individual differences in vulnerability to this neural sensitization, but some individuals are especially vulnerable. And of course, what it meant was that if some individuals could sensitize their dopamine system, and if dopamine was wanting, then wanting would go up and up in a sensitized addict, even if liking went down. Well, that was a theory posed in the 90s, and it's probably fair to say that virtually nobody believed that theory for about at least the first 10 years of its life. We believed it gradually, but there weren't many others who did. One of the reasons that more of the world in the last 10 years especially has come to perhaps believe the theory is because of a diabolical experiment that nobody would ever ethically approve, but which medical science has inadvertently accomplished. And it's the invention of the new medications in the last 10 to 15 years, the new medications for treating Parkinson's. The older medications are drugs like L-Dopa. L-Dopa causes the brain to make natural dopamine. Even neurons that couldn't make dopamine, when you take L-Dopa, the neurons can make natural dopamine. That's been around since the 70s. But the new medications for dopamine for Parkinson's disease are essentially fake dopamine. They're direct agonists, direct dopamine agonists that can mimic dopamine at especially particular kinds of receptors, D2 and D3 receptors, particular kinds of dopamine receptors in the brain. They mimic the dopamine. They don't need natural dopamine. This is a marvelous, marvelous discovery as a treatment for Parkinson's disease because it helps a lot of people. But at least 15% of the patients who take the direct agonist medications begin to experience strong motivations that can essentially become addictions. They can be compulsive gambling, compulsive sex, compulsive shopping, compulsive binge eating, compulsive hobbies or punding. Punding is like sorting buttons in a drawer. Fortunately, if it happens, you can reduce the dose and often these things go away. But what it's really doing is taking individuals who are not addicts who were least likely ever in their life to become addicts, and making them into addicts by giving them dopamine stimulation. And it's not incredibly pleasant, these drugs. It's not that it's an incredibly liked effect. It's a essentially wanting effect. And there's evidence that in the particular patients, the 15% who develop the compulsive addictive motivations, what's happening is that they have a sensitized dopamine system, just the ones who are developing the compulsive addictions. This is showing you dopamine release, bars of dopamine release in parts of the striatum, including the nucleus accumbens. And what you're seeing is dopamine release being elicited by a drug of L-Dopa, a dose of L-Dopa, which causes natural dopamine release. It's being given to Parkinson's patients here. Both the blue bars and the red bars are Parkinson's patients. Everybody's getting the same L-Dopa. But you can see the blue bars are higher. There's more dopamine release. Who are they? The blue bars are the Parkinson's patients who have the compulsive motivations. The red bars are Parkinson's patients taking the same drug, but who don't have the compulsive motivations. The ones who are sensitized, showing extra dopamine release, they have the compulsive motivations. They've been sensitized by their medication, and they're showing you the sensitized brain response, and the addictions are the sensitized psychological response. So essentially, a human experiment inadvertently created by medical science. That kind of evidence has led more people to the notion that dopamine systems are wanting, and that this large green brain system centered on dopamine but extending to all of these sites, that this can cause powerful wants. Still, for my colleagues and for me, for the first 10 years or so of our hypothesis, it was a little bit uncomfortable. And my colleagues would often say at audiences and at meetings, okay, well, if dopamine isn't pleasure, then what is pleasure in the brain? Where are the pleasure mechanisms if dopamine isn't? And if pleasure electrodes aren't necessarily pleasure, what is causing pleasure? Even we eventually could find pleasure bases. So these little red hot spots here, these are pleasure mechanisms that can cause true liking, that can generate true liking, even in our hands in rat experiments, using the kinds of liking reactions to sugar, the taste reactivity experiments that Fred described. Each little red spot is a hedonic hotspot that has the capacity to double or triple liking, to intensify pleasure for sweetness and other rewards. But never dopamine. Dopamine can never do it. Opioids, natural heroin in the brain, can do it. Endocannabinoids, natural marijuana neurotransmitters in the brain, can do it, but never dopamine. The liking circuit, though, is fragile. The wanting circuit is easy to turn on and it's robust. The liking circuit can only be turned on by a few neurotransmitters. It's anatomically tiny, each of these hotspots. I'll show you an example. And you need unanimous activation of all the hotspots together in order to enhance liking. Let's see what a hotspot looks like if we look close up. This is the nucleus accumbens hotspot here, and we've blown it up here. It's blown up. This blow up, it looks like a little bit like a neuroimaging experiment, as though it were neural activity, but it's not a neural imaging experiment. This is a neural causation experiment of psychology, of psychological processes. It's a neural mechanism causing psychological processes. Each splotch that you see, whether it's red or green, each splotch is a tiny microinjection, a tiny droplet of opioid-stimulating drug put into the nucleus accumbens painlessly in the rats. And we can see the splotch by measuring neural activation. If you look under a microscope, you can see little green specks of activated neurons. They're basically generating phosphoprotein. Their genes are activated by the opioid, and they are generating phosphoprotein within them. When they're activated, it tells us how large the splotch is. That gives us the size of each splotch. But the color of each splotch is the psychological consequence of what this opioid stimulation is doing. Wherever it's green, every green splotch, and the green extends under the orange, so the green goes out here. Wherever it's green, it's increasing wanting to eat. The opioid stimulates wanting to eat. It doubles the amount of food the rat eats, or it quadruples the amount of food the rat eats, or it makes the rat eat 8 times more than normal the amount of food. So very intense appetite. And the green, as I say, goes under the splotch, the red splotch. The green would come out into the room here, fill up the nucleus accumbens. It would fill up other parts of the brain around the nucleus accumbens. Massive green, triggering wanting to eat. But the red splotch is a cluster. It's a whisper from the universe to us. It's telling us that all the splotches that do a second thing, that actually also increase liking reactions to the sugar, double or triple the liking reactions to a sugar taste, they all tended to cluster. There's no reason why liking spots ever had to cluster in the nucleus accumbens, but they do. That's what the brain is telling us. They're all clustered, and this defines the hedonic hotspot. So that's a hedonic hotspot. It's the one part. It's only 1/3 of the nucleus accumbens medial shell here. It's about 1/10 of the entire nucleus accumbens. Tiny. But it has this capacity to amplify liking. There's a hotspot there in the accumbens. There's also hedonic hotspots in the cortex. This is a rat cortex. We're seeing the lateral surface of the cortex here and the medial surface of the cortex here, the midline surface. And wherever it's red, you can see liking splotches. There's a hedonic hotspot in the very front, both in the side and in the midline. This is one hedonic hotspot here. The orbital frontal cortex has a hedonic hotspot that's both on the side and the midline in the middle. And then there's a second hedonic hotspot in the insula, further down along the side in the cortex. These are hedonic hotspots in the cortex too. I said that you need unanimity of the hotspots to cause liking, and that's what this slide is telling us. This slide is showing us a microinjection just in the orbital frontal cortex, in the orbital frontal cortex hotspot right here. It's a microinjection of opioid-stimulating drug, Dango right there. And what the slide is telling us is that the microinjection of opioid stimulation here is also triggering brain activation, reflected as FOS increases in the nucleus accumbens hotspot right there, and in the insula hotspot on the lateral surface of the cortex, and in other hotspots down in other parts of the brain too. Turning on one hotspot turns on the others. The first hotspot you stimulate, it recruits the other hotspots. They come on unanimously. And if we were to turn one on but suppress a different one, we would not get any enhancement of liking. If we turn one on and allow it to recruit the others, then we get the doubling of liking, the tripling of liking, the pleasure is enhanced. It's a unanimous network that operates as a whole, and it has to be unanimous. So those are the hedonic hotspots. They are a basis of liking, but because they're fragile functionally, hard to turn on, easy to disrupt, it may be a reason why intense pleasures are relatively few in our lives and spaced apart compared to intense desires. It's wanting is simply easier to turn on. So that's sort of a recap of the wanting and liking. More recently, we've been trying to get a better handle on wanting, the psychology of wanting, the incentive salience principles, the Bindra-Totes motivation theory, how it operates, creating even wants for what hurts you, as I'll show you in a moment, but also, in a sense, trying to get a better understanding of the focus of wanting. If you turn on a pleasure electrode the first time, it may or may not be real pleasure, but it makes the world attractive. Even humans who have these electrode turned on, the whole world brightens up. Individuals around them become interesting, maybe romantically attractive, who, if they weren't necessarily before. The world is inviting when it's turned on. But in natural life, what you want at this moment is different from what you wanted at another moment. The focus of your wanting switches from food, water, to other things. And in addiction, the focus of wanting becomes sort of narrowed and fixed on one thing above all. What's controlling this focus? That's what we're trying to understand. To get a handle on this, I'll show you some recent experiments from our lab. We're using a different technique, and we're going to interact with the brain dopamine system through the amygdala. We're going to use the amygdala to control the brain dopamine system. When we look at things around us, recognize their emotional and motivational importance. It's partly the amygdala that's helping us to do that. So that's why we're going to do the look at the amygdala. To turn on the amygdala, we're going to use some fairly recently developed techniques, optogenetic stimulation of the brain, developed by Carl Deisseroth and others about 10 years ago, using laser light to naturally activate neurons by putting photoreceptors painlessly into the brain so that natural laser light can turn on the neurons. Let's give you a sense of how that's working. These are the rats who have this, and you can see when the laser is turned on, it's a blue laser light. The rat looks around. It's not bothering the rat in any way. It's not at all painful. But what it's going to do is amplify a particular psychological function. This is how the rat looks when it's not connected to it. It basically doesn't know that it has the head cap. It can walk around and do other things. There's no pain here if you touch it. There's no pain there. But you can see the little optic fiber that the laser can be connected to send laser light down into the brain. What happens to turn this on is we're going to see the rat has... I thought I had one more slide. Oh yes, here we go. So to turn this on, to see how this is going to work, you know, the rat was anesthetized, and a tiny droplet of a virus solution was placed into its amygdala. This virus solution contains a gene for a photoreceptor. The virus cannot replicate. It's not like the coronavirus, it's not like the flu virus that can replicate throughout a body or jump. This virus can only infect a neuron where you place it, so it cannot spread. But the virus is carrying a gene for photoreceptor. In our eyes, we have photoreceptors. When you look at the light, the photoreceptor molecules change shape as photons of light strike the photoreceptor molecule. The molecule changes shape, and it allows ions to flow into neurons and eventually stimulate neurons so they fire. This photoreceptor, coated by the gene, if it infects the neuron will make photoreceptors. The neuron will sprout photoreceptors. And if light is shown on the photoreceptor, the neuron, the molecule will change shape. It'll allow exciting ions to enter the neuron, and the neuron will fire. It will be excited. It's entirely painless. We can even ask the rat if it doesn't want to have it turned on, and we'll see the rat doesn't really care if it's turned on or not. But we'll see what it does psychologically to the rat. And the blue laser light will make the neuron fire. Now the rat that we're going to see, let's go back to the slide. This rat has a happy choice. This rat can press a lever and it'll earn a sugar pellet. Or the rat can press a different lever and earn essentially the same sugar pellet, but also have her amygdala stimulated. And we'll see what it looks like. Let's just actually see what this looks like in a little video. Here we go. This is Mike Robinson and Shelley Warlow. So here's the rat. At this point, the rat's connected to a cable, so she can't bite through the light. The lever comes out of the wall, and she pressed on the lever, and she earned a sugar pellet that was right there. This is where the sugar pellet was. She's gone back to the lever, but it's gone away. Now the other lever will come out, and we'll see what she does. She seems very eager and interested in that too. She's biting on the lever, pressing on the lever. She's earned the sugar pellet, the second sugar pellet. She got the sugar pellet from here. So she's interested in both of those. Sugar. Let's give her a choice, both levers together. Both levers come out. She goes to the laser lever, earns the sugar and the laser. The sound tells her she's earned it. There's a little timeout period. She cannot earn another sugar for 8 seconds or so, but she's just working away on this lever to try to get through that timeout. She's got through the timeout. She's earned a third sugar pellet. She's ignoring this other lever. She could earn more sugars by going back and forth, but she's not doing that. She's staying fixed on her laser lever. Give her one more chance. She's earned that sugar. Oh, she looked at this lever, but she's gone back to the laser. And she just will stay utterly obsessed and focused on the laser lever. She will ignore the alternative sugar, even though she could earn more sugars if she went back and forth. Why does she so much want this laser sugar or laser lever? Is it that she wants the laser itself? Is the laser a reward in the amygdala? Is it a pleasure laser in the amygdala? Or is the laser perhaps making her want that sugar more because it makes her like the sugar more if she has the laser on when she's eating the sugar? Either of those could be possible answers. We can ask her both questions. Let's ask her, does she want the laser itself? Is that what she's going for? We can ask her by telling her, if you touch an object here, you'll get the laser, only the laser, no sugar. Touch this object, every time you touch it, you get the laser. Touch this other object, you get nothing. She touches the laser object a few times. She touches the other object that gives her nothing a few times, the same number of times, and then she ignores them both, just a couple of times on both, and then ignores them. She doesn't seem to care about the laser. Well, we could ask her again. We could ask her the way Jim Olds originally discovered pleasure electrodes. How did he do that? How did he discover pleasure electrodes in rats? He put the rat, the very first rat who had the very first electrode in its hypothalamus, on the table. And whenever the rat went to one corner, he turned the electrode on. And in other places, he did not turn it on. And he found that the rat was staying all the time in the corner that got the electrode on. This was a measure of the reward of the electrode. We can ask the rat the same thing in the laser chamber. We can put her in and let her wander around the corners of the chamber. Each circle tells you the percentage of time, the amount of time she spends in that corner, the size of the circle. The blue is the one corner that has the laser. And basically the circles are all equal. She does not seek out the laser corner. Unlike the electrode rat, she does not seek out the electrode corner. She does not avoid the laser corner. It's not that the laser in any way distresses her or repulses her or that she wishes to not have it turned on. She doesn't avoid it. She just doesn't care. She wanders randomly. The laser by itself is nothing to her. But put the laser on a sugar, and that sugar becomes tremendously valuable. If we put the laser on cocaine, we can ask her before we leave this, did she then want the laser sugar more because the laser made her like the sugar more? We can ask her with the taste reactivity test that Fred described. Let's actually look and see what the taste reactivity test looks like. Let's see a human newborn infant on the first day of life. This is Jacob Steiner in Israel giving sugar water to a human infant. Here's the human infant. Just got a droplet of sugar water. Is sort of showing relaxed tongue protrusions. That was fast. Let's see it one more time. Relaxed face and tongue licking to the sugar. This rat is receiving painless sugar water, painlessly infused into his mouth through oral cannula, permanently implanted oral cannula, utterly painless. And he's just responding to the sweetness, to the sugar, licking the lips. Here's a bitterness, quinine taste, bitter. A little bit different. Gapes of the mouth, triangular gapes, scrunching of the eyes and brow. The rat cannot scrunch its eyes and brow, doesn't have the musculature, but the rat, he can gape, he can shake his head, he can shake his paws, he can rub his chin on the classic floor, and he'll do all of those things, gape to it. If we ask, does the laser change the perception of the sugar taste? Here's the answer. These are the positive liking reactions in the presence of the laser, when sugar is being given in the presence of laser. Here's the liking reactions to the same sugar in the absence of laser, and basically there's just no difference. The laser is not changing the liking or disliking of the taste. It doesn't change it at all. It makes the sugar wanted, but it doesn't make the sugar more liked. It's a bit like a dopamine manipulation. What if we put the laser on cocaine, intravenous cocaine? Here's another rat who has the laser cap for the amygdala stimulation. She also has an intravenous cannula that allows her to painlessly receive intravenous cocaine if she wishes to do so. If you hook the cannula up to a cocaine solution and let her press a lever or poke her nose into a nose hole to earn cocaine, let's see what happens. We'll give her a choice again. She can poke her nose into this hole and earn just cocaine, intravenous cocaine, or she can poke her nose into this hole and earn cocaine plus laser activation of her amygdala. This is the cocaine-only porthole that comes out. She pokes into it, and she's now earning cocaine coming in through here. That's the jugular cannula. Now the other porthole comes out. She presses into that and gets laser activation, but also cocaine. You can see now she's a little bit excited and hyper. She's showing a little cocaine stereotypy, so she's a little bit hyper. She looked at that one, she looked at this one when she has the choice. She hasn't triggered either, but now she triggers that one, and she will stay fixed on that one, all the way on that one. She'll ignore the other one. She'll basically stay utterly focused on the laser cocaine until the end of the session. Again, this rat will not self-stimulate laser alone. She does not seem to care about the laser alone, but it makes that cocaine tremendously valuable. Suppose we pit sugar against cocaine. Give you a choice between sugar or cocaine. Which would you prefer? We're going to need two groups of rats for this. For one group of rat will have the laser placed on cocaine, but can earn sugar without laser. The second group of rats will have the laser placed on the sugar, but can earn cocaine without the laser. What happens? The rats who had the laser on the sugar work only for the laser sugar, and they ignore intravenous cocaine. There's nothing magic about intravenous cocaine. Pitted against sugar here, it becomes tremendous, the sugar wins. But the rats who had the laser placed on cocaine work only for the cocaine, and they ignore the sugar. If you're wondering what would ordinary rats do in this situation, here's the control rats. It's 50-50. We adjusted the sugar and cocaine doses to be about 50-50, but it's showing you the power of the laser. Again, whether it's sugar or cocaine that it's making them pursue, most of these rats will not self-stimulate the laser alone. It makes that sugar tremendously valuable, that cocaine tremendously valuable, but by itself, the laser is not so valuable. What if we, last experiment, what if we pasted the laser on something nasty? So far it's been on nice cocaine, nice sugar. What if we did it on something nasty? This will be a situation where a rat could receive an electric shock, but it does not have to. It will only be voluntary if it does so. The shock rod is in the chamber. Here's the shock rod in the chamber. And if the rat touches the shock rod, it will receive an electric shock because it's electrified to give you a mild reaction. You can stand it if you put your hands on it, but it's not pleasant and you will jerk away. You'll see what a normal rat does. A normal rat, when it touches it, will touch it maybe once, maybe twice, but then it will never touch again. It will stay far away and will actually begin to show an anti-predator reaction, burying the rod. If there's sand or sawdust in the chamber, it will bury the rod. That's what this ground squirrel here is doing in the California Sierra Mountains to this rattlesnake. This is a rattlesnake who's approaching the burrow of a mother ground squirrel in the spring. In the spring, mother ground squirrels have pups, squirrel pups, in their ground burrows. The rattlesnake cannot eat the mother because the rattlesnake cannot swallow the mother. But the rattlesnake could eat the pups, so the rattlesnake wants to get into the burrow, and the mother is defending her burrow by kicking sand into the face with this anti-predator reaction. Sometimes the mother will be joined by sisters or daughters or her mother, matrilineal relatives who help. But you can imagine that's a fearful, frightening situation for any ground squirrel, because ground squirrels can be bitten, even if not eaten. What does the rat do? Let's see a normal rat and then the laser rat. This is a normal rat. Here's the shock rod. Got a shock. She touched it. She got a second shock. She will never touch it again. This rat, she's staying far away now, and she's beginning to defensively. Here's a laser rat. Touches the shock rod. Gets a shock, but goes back to the rod. Seems very fascinated by this rod that just shocked her. Fascinated, fascinated. Almost holds her paw back, but she will get another shock, and she will get another shock, and another shock, and another shock. She will be fascinated by the rod. If you leave them in for 5 minutes, she will probably receive at least 5 shocks. We take them out after they've had 5 or 7 shocks, because why go on? But I imagine that she would go on and continue to shock herself. It's actually the rod giving her the shock that's part of the fascination. If the rod were not electrified, she would ignore it. It would be like self-stimulation. She would just not approach the rod. But if the rod gives her the shock and the laser is paired on that, the rod becomes irresistibly fascinated to the point where she will touch it again and again. Now, it isn't, I think it is not that she wants the shock. What she wants is the rod. That is the associated cue for the shock. She's fascinated by the rod. Why do I think that she wants the rod? This is a Bindley-Lipposian distinction. It's the cue. Why do I think that she wants the rod as a cue? Well, we can ask her, because there's another cue too, and we can ask her about what she thinks about cues for shock. Every time the rat touched the rod and got a shock, it also heard a particular sound, beep. Whether it was a laser rat or a normal rat, it heard beep whenever it got a shock. Now you can ask a rat, would you like to hear a sound that's associated with shock, that's been paired with shock in the past? And normal rats will tell you always, absolutely not. I do not want to hear any shock-associated sounds. You can ask the rat in an instrumental conditioned reinforcement test where you put the rat in a new situation. This rat has never seen portholes before. But if she pokes into this porthole, she'll hear the sound, just the sound, beep, that means shock. If she pokes into here, she'll hear a different sound that she heard in her home cage. What do rats do in that situation? The gray bars are normal rats. Let's look at them first. The gray bar is a normal rat who says, no, I do not want to hear the shock-associated sound. They're actually below 0 on this test, for this normalized test. They avoid the shock-associated sound. They do want to hear a few times the other sound that they heard in their home cage, but they do not want to hear the shock-associated sound. A laser-paired rat who was attracted to the rod wants to hear the shock sound, will work to hear 15 presentations. This is only a 15-minute test, but this rat will hear 15 presentations of this shock-associated sound. Give me the shock sound again. Again, that shock sound again. This is probably the first rat in the history of all rats in 65 million years who has ever wanted to hear a shock-associated sound. But that's what this laser does. It makes the cues for the shock attractive, just as it made the cues for the sugar attractive or the cues for the cocaine attractive. You might have noticed the rats were nibbling on the portholes for the sugar and the cocaine. At the moment, how is it doing it? How is it making shock-associated cues attractive? Because at the moment that the rat encounters that shock rod, the rat who was attracted, it lights up the entire mesolimbic motivational system that lit up in the neuroimaging slides I showed you at the beginning, in people who were listening to favorite music, cocaine, and looking at sex, pornography things. It's lighting up the dopamine neurons in the ventral tegmentum. It's lighting up the nucleus accumbens, especially in the hedonic hotspot. It's lighting up the orbital frontal cortex and the posterior insula that have the hedonic hotspots. It's lighting up that incentive motivation system that powers the Binder-Totzian motives. So this is a laser that essentially jumps into the brain and makes cues for nice things or nasty things incredibly potent Bindra-Tots wanted events, able to trigger motivation strong enough to drive an individual to receive repeated shocks, and the kind of motivation that could be potent in addiction. The very last thing I'll say about this, though, and it kind of relates to the pain side of the title of this Agony and Ecstasy theme, and it relates to Fred's mentioning in the beginning that there's a dark side to dopamine. This system can be turned into a fear potentiating system rather than an irresistible desire system. The same laser that I just showed you, in the same rat that I just showed you, who was attracted to her shock rod, that same laser can actually enhance fear in a different situation. So if we went to a traditional Pavlovian fear learning situation where the shock is not controllable, it's not assigned to just the rod, what happens is the laser, the same laser, actually increases fear reactions of freezing and avoidance of the cues. There's such a thing as incentive salience. That's what dopamine is famous for, perhaps. But there is such a thing as fearful salience, which is also dopamine driven in the striatal system. That's relevant to paranoia and things. We'll see it in a moment. And that's what this can do. The amygdala and the dopamine system can flip its valence, which is a major thing to understand, a thing that we do not really understand how it flips its valence from desire to fear. But it's, I think, a challenge in the next few years to try to understand. So the very last thing I'd like to mention are just some clinical applications of these ideas. There isn't really a strong application yet of the shock rod attraction because that's brand new. Although one can imagine that it is very involved in addictive wants, the same want. All the shock rod is showing you is the want can happen in the absence of any liking at all. The shock rod could also be relevant to things like self-harming, self-cutting, perhaps. Is it possible that some acts of pain actually become attractive, the act of them, the cues of them, the act of carrying them out? Is it possible? This doesn't say that that's happening, but this raises the possibility of saying the brain has a mechanism that could produce that sort of thing. But the applications that exist, of course, drug addiction was the original application with Terry Robinson, the notion that the drug mesolimbic dopamine hyperreactivity causes potent cue-triggered wanting, surprises, urges triggered by cues, but the power of the urge can be modulated by your state at the moment. So stress, for example, will enhance that. What is it like to be a sensitized addict? What is that temptation? What is it? What is that compulsion? In what sense is it compulsive? My belief is that this compulsion of a sensitized addict is somewhat akin to the compulsion that any of us could feel for food if we were starved enough. If we were starved enough, people begin to dream about food, they begin to find food compulsive in a number of ways that the rest of us would be able to resist. It is possible to resist even through starvation, but it's a challenge that most of us are not called upon ever to face in our lives. And it's possible that a sensitized addict is. That's the nature of, if there is a compulsion to addiction, I think that's the nature, the surprising power of some occasions of cue-triggered urges in a sensitized brain. As I say, nobody believed the theory throughout the 90s or much of the 2000s, but in the last 10 years it's sort of become more highly cited. Why is it more highly cited? Partly things like the Parkinson's patients I told you about becoming addicts, partly other things including experiments that we and others have done. But basically, if we go back to human addicts looking at drug-related cues, the first slide that I showed you, it was showing you this, and this is the addict who's showing brighter lighting up of the brain. What is this? This is the amygdala, the same site where our laser stimulation was placed in the rats. This is the human cocaine addict lighting up especially. That's a sensitized brain response. Hyperreactivity to the appropriate cue, to the addictive cue, the relevant cue. 10 years ago, I would have said that drug addiction is the only form of addiction that incentive salience is relevant to. And that if there are other behavioral addictions, they're not addictions in the same sense or same way. I now think I would have been wrong 10 years ago to say that. Because in the past 10 years, there's been a number of neuroimaging experiments in humans that have changed my mind. Basically, they're showing the same brain signature in people who are showing, at least some people who are showing compulsive eating, or some people who are showing compulsive gambling or compulsive sex addiction. What is the sensitization signature, the brain signature? It's cue-triggered hyperreactivity, triggered by the relevant cues. Food cues for compulsive eaters, gambling cues for compulsive gamblers, sex cues specifically. Hyperreactivity. Hyperreactivity in what way? Two ways. Hyperreactivity in the sense that that person shows a higher reactivity to the, say, drug-paired cues than to winning money or to nature cues or to other cues, just as this was showing us here. This is the same person showing extra reactivity to just the drug-paired cues. So hyperreactivity to the relevant cues, but also hyperreactivity in a second sense. Hyperreactivity in the sense that lighting up to the addictive cues more than the rest of us would light up to the same cues, even if we also eat and are tempted by tempting palatable foods, as we all are. Even if we also gamble recreationally. Even if we're interested in sex too. Hyperreactivity in the sex addict or the gambling addict above and beyond the level triggered in other individuals by the same cue. That would be the sort of powerful temptation, the starvation-like temptation power in a non-starved but sensitized brain. This idea has been applied not by me or my colleagues, but by people doing the human neuroimaging experiments. So food addiction is a label Ashley Gerhardt and others have applied to the people who are showing the sensitization signature. Hypersexual cue reactivity in putative sex addicts who pursue pornography and masturbation throughout the day to the extent that even they are distressed. Gambling, suggested by these sorts of papers. Are these true applications? I don't have an opinion. But to the extent that this signature, this dual hyperreactivity signature, is showing up in these individuals, to that extent I would say that's a plausible case for food addiction or gambling addiction or whatever addiction it is to be. How many individuals will have that signature? I have no idea. It could be a small proportion of those who pursue, but that would be an interesting question that would have to be. The dark side beyond addictions, beyond. Fred had mentioned that in depression there could be a loss of wanting without liking. This is a well-studied phenomenon now. It's not just in depression, but it's also been reported for other conditions that have, for decades, been claimed to have also anhedonia. Anhedonia is the textbook psychiatry term that was applied to depression. Anhedonia of depression meant you couldn't have pleasures in life. Anhedonia in Parkinson's disease, when not medicated, the person who had no dopamine, anhedonia has often been associated with Parkinson's disease, would mean no pleasure in the absence of dopamine. Anhedonia in some forms of schizophrenia. Textbooks in psychiatry have anhedonia for all three of those conditions. But all three of those conditions, a number of researchers are now reporting that rather than be true anhedonia, loss of pleasure, it's really more like loss of dopamine, wanting, even if pleasure is still there. So if you give these people tasty ice creams, they still like them as much, but neither the ice creams nor anything else in life have motivating value that's not worth living for in these kinds of conditions. So new terms have been suggested, rather than anhedonia, terms like avolition, loss of wanting, of volition, without loss of hedonia, pleasure, or motivational anhedonia, as opposed to true or consumatory anhedonia, true loss of pleasure. Are these true distinctions and applications? Again, it's not for me to say, but they are possibilities, and some data is out there to suggest that they may apply. And the very last sort of clinical application, the true dark side of dopamine-related systems, is the possibility of fearful salience in some forms of paranoia. This is an idea that's been championed mainly by Kshitij Kapoor, a psychiatrist who used to be here in London, but now I think is in Australia, and his colleagues, a number of whom are still in London. This is an idea that schizophrenics who have paranoia may have hyperreactivity in their nucleus accumbens and striatal dopamine systems, but it's not necessarily desire or incentive salience, it's more of a fearful salience. It's the kind of thing that can operate if someone were to step outside and see a police car. If any of us stepped outside and saw a police car, we might feel more safe, quite conceivably. But someone with this state who steps outside and sees a police car might think, I saw a police car yesterday also, and here's a police car again. The police are following me, and this is part of the world being against me. It's always been a truism in psychiatry that if you gave neuroleptic antipsychotic drugs, medications for schizophrenia, it takes 2 weeks or so for the effects to kick in. But Kapoor and colleagues have argued, no, that's not the case. If you give these neuroleptic drugs, which are dopamine blocking drugs, they're D2 receptor blocking, dopamine receptor blocking drugs. Even the new atypical antipsychotics, which also block serotonin receptors, they still also block dopamine receptors. And Kapoor has argued that the efficacy of all of these drugs is really best correlated with their dopamine blocking rather than their serotonin blocking. Kapoor and his colleagues argue if you give these drugs to a patient on the first day even, you can detect an effect. But it isn't that the delusions have gone away, the delusions that the world might be against them. It's rather that they can look at the police car and say, yes, the police are following me, but it doesn't bother me quite so much today as it did yesterday. And if they take the drugs, the neuroleptics again and again, it gives them sort of a motivational distancing. The motivational power of those cues declines. The cognitive delusions are separable. But if you have that motivational space where these are no longer compelling, then there may be a space for the delusions to dissipate by other means. The delusions are not dopamine dependent, but the motivational cue-triggered fearful salience may be. That's the notion. So I will stop. All I've said is that from Bindra and Totes, the principles of this system, cues as powerful motivational magnets, cues as trigger for motivation, we can see that reward is a configuration. Things like fear may also be a configuration that can be separated into different psychological components. Addictions in particular do seem to arise from the hyperresponsive wanting system. That system can flip its elements to magnify fear and paranoia. But the last thing I would say is all of these experiments, even the brain manipulation experiments, even in rats, for me they have one major goal. The goal, of course, is, yes, to understand how brain systems work a bit better, but the major goal is really to understand how the psychological systems, how the Bindra-Totes theory works better. And it's sometimes we get these new insights. Insights like it's possible to want even what hurts you. It's possible to want in the absence of any liking at all. Insights that might have applications to clinical situations in life too. So it's possible, I think there'll be more surprises, more disappointments, but also more insights in the future as well. I haven't done any of the work that I've described. It's all been done by other people. I just tell you about it. These are the ones who do it all. And thank you for your attention.